Hello, and welcome to this CADASIL Care webcast. The CADASIL Care series has been developed to address issues of importance to those diagnosed with or at risk for CADASIL and their families. This webcast is hosted by cureCADASIL, a nonprofit 501c3 organization dedicated to finding a cure for this rare genetic disease. This and other webcasts are available on our website at curecadasil.org. Based on survey results from the CADASIL community earlier this year, we will be talking to various experts in the field of CADASIL, bringing up topics of interest from patients and families. My name is Janet Mills, and I'm a CADASIL patient and advocate for cureCADASIL since 2012. Today, I am very pleased to introduce Dr. Michael D. Geschwind from University of California at San Francisco. He will be sharing information about the diagnosis and management of CADASIL. Dr. Geschwind is a professor of neurology at the University of California, San Francisco. His primary areas of clinical research are in rapidly progressive dementias, Huntington's disease, spinocerebellar ataxias, and atypical Parkinsonian disorders. He and his team run an active clinical research program in sporadic and genetic prion disorders. Dr. Geschwind helped run the first ever treatment trial for sporadic Jacob-Creutzfeldt disease, SJCD, in the United States. Over the past 16 years, he has helped recruit one of the largest clinical cohorts in the United States of patients with prion disease, as well as patients with other non-prion RPDs. Dr. Geschwind also is involved clinically in caring for patients with mild cognitive impairment, Alzheimer's disease, frontotemporal dementia, Huntington's disease, CADASIL, and other adult-onset leukoencephalopathies, atypical Parkinsonian dementias, and various neurogenic disorders. Thank you so much, Dr. Geschwind, for your time today.
Thank you, Janet, for that kind introduction. I'm sorry that I gave you too much to say, but I thought you did quite well with a lot of those complicated words. A lot of the neurologic diseases, we come up with the most complicated names for them, and it's nice to have a shortened name, CADASIL, for the disease we're going to talk about today. I should mention that I was involved in the first treatment trial for CADASIL back in the early 2000s that occurred primarily in the United States, and that's actually sort of what spurred my interest in this disorder. I'm generally interested in many neurogenetic diseases, genetic diseases that affect the brain, particularly in adulthood. So the outline for my talk today, I'm going to give a background of CADASIL, try to define it, present a little bit of the history, a little bit on the demographics, and then I'm going to talk about how we make a diagnosis of CADASIL. We'll talk about the family history, some of the classic signs and symptoms. Symptoms are what patients report, signs are what a physician or a neurologist finds on their examination of a patient. We'll talk about how the brain imaging, the brain MRI, can be very helpful for diagnosis, and some of the other aspects of the disease, skin biopsy, genetic testing, and I'll talk a little bit about the diagnostic algorithm for working up patients who have diseases of the white matter as an adult, or leukodystrophies as an adult. And we'll talk about management of the various symptoms of CADASIL and some of the studies that are ongoing and that might be coming up. So CADASIL stands for cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy. Really a mouthful. And I'd like to break it down to explain why, what this term means. So cerebral is a term that means the brain, and CADASIL only affects the brain clinically. We don't know why that is, but it's the only organ in which there's actually damage that we can see clinically. The skin, the blood vessels in the skin are affected, but we don't see a skin disease. We don't see heart disease. The other organs in the body are healthy. It's the blood vessels of the brain that are unhealthy, and it's not clear to researchers exactly why this is, but it preferentially affects the brain, hence the term cerebral. Autosomal dominant. So I'll get more into this term later, but suffice it to say for now that it means it's a familial disease. It's a genetic disease. It runs in families. Arteriopathy. So this is a term, arterio, are small vessels in the body. And in this case, they're small blood vessels in the brain. And pathy means there's an abnormality. So there's an abnormality in the small vessels in the brain. And in this disease, it affects the vessel wall. So think of a pipe, and the wall of the pipe becomes extra thick. And the lumen of the pipe, or the opening of the pipe, is actually, it becomes smaller only because the walls become thicker, and it therefore narrows the lumen, or the opening of the pipe. But unlike typical cerebrovascular disease you might see from somebody who has diabetes, high cholesterol, high blood pressure, where we actually see plaques forming, deposits of fat or other material along the inside of the wall of the vessel, that doesn't usually happen in CADASIL. The inside of the lumen is actually clear. It just becomes smaller and smaller because the wall becomes thicker because of the deposition of some material in the wall that causes it to thicken. And the vessel wall becomes easier to break, becomes more friable, which is why you can also have bleeds in the brain from this disease. And then the next term, subcortical infarcts. So subcortical refers to a part of the brain. The surface of the brain is called the cortex, and the surface of the brain has some gray matter. It's 6 layers of nerve cells. And then underneath that, there is connections of those nerve cells on the surface of the brain to collections of nerve cells in the center of the brain. Those connections are like the wiring of the brain. And the wiring of the brain, those wires, are what we call axons, are surrounded by insulation. And in the brain, the insulation happens to be fat, and fat is white. When you actually take somebody's brain when they're done with it and open and look at it, cut the brain, the inside of the brain, most of it is white because you're looking at fat surrounding blood vessels, and fat surrounding the axons. And that area, it looks white, and therefore we call it white matter. And that's the area that's subcortical, and that's where the strokes occur, or infarcts is the medical term for strokes that occur. And then there's the last term, leukoencephalopathy. Leuko means white, and white matter is the area, the axons that are covered in fat. I've described why we call it white matter. And that's where the disease seems to, where we see the disease, and we can particularly see it on brain imaging, brain MRI sequences. And then encephalopathy. Encephalo means brain, and pathy or pathology means abnormal. So the brain is abnormal because of the problem of strokes in the white matter. And the strokes occur because the blood vessels become too narrow. So in history, a lot of the literature says that the first descriptions thought to be CADASIL were by a Swedish team of doctors, Sarander and Wallander, in 1977 in the eminent journal The Lancet. And there they called it hereditary multi-infarct dementia. The same year, they published a much more detailed study of the same family with, I believe, 5 affected members. And in the second paper published in Acta Neuropathologica, another esteemed journal, they went into much more detail, showing the detailed brain pathology of these patients and much more detail about their history. It turns out that another family was described the same later that same year in England, and this was published in the same journal, Lancet. And the title of the paper was called Chronic Familial Vascular Encephalopathy. Chronic meaning long-term, familial meaning the disease seemed to be present in the family, and it affected the blood, the vasculature, the vessels, and it caused encephalopathy. And they started the paper by saying the condition which Dr. Surander and Wallander call hereditary multi-infarct dementia seems to resemble the disorder that we have described. And they described, but they hadn't published it yet in the literature. They published it as abstracts and presented it at meetings, and that's what those citations 1 and 2 are for. And they said a detailed account of this disorder, which we call chronic familial vascular encephalopathy, will be published elsewhere, but they basically summarized that it was an autosomal dominant inheritance, which I'll go to in more detail what that means. The age of onset was around 50 years old, actually about 10 to 20 years later than the paper by Surander and Wallander. And later, initially patients had strokes, and then they had transient, or maybe what we would call temporary, motor symptoms, so trouble with movement, sensory symptoms, numbness, tingling, and then other symptoms. And later, these patients developed dementia, trouble speaking, trouble swallowing, difficulty moving their limbs, walking, urinary incontinence. And they questioned in this paper whether it was the same condition that had been published by Surander and Wallander, but they noted that there were some differences, and they wondered whether it was the same condition or not. And it turns out that actually in 1955, there were 4 Belgian sisters described in France with a severe cerebrovascular disease. And many people think that these might have been the first cases. So we have the Swedish family that I mentioned in 1977, an English family. And then in 1993, a French group coined the term the CADASIL, and in describing several French families that suffered from migraine attacks and recurrent strokes. This was published in 1993. 3 years later, the gene was discovered by a similar French group. And then in 2007, a paper came out that actually determined that the Swedish family from Surander and Wallander actually did not have the same gene. It's actually a different genetic disease to which we still don't know the mutation, to my knowledge, but it's not CADASIL. And clinically and radiologically, meaning on brain imaging, there are some differences. But there are several other diseases that sometimes can mimic CADASIL, but are slightly different. So that's sort of the history of the discovery and the identification of CADASIL. It's a disease that occurs worldwide, multiple different ethnic groups. More than 300 families have been identified, and this keeps growing. There are probably many more cases than we know about. I think we're somewhat at the tip of the iceberg with this disease in terms of there are far more cases out there, I suspect, than we are aware of. The largest number of families have been identified in Europe, and perhaps that's just because many of these cases were first described. And at least 160 mutations have been identified in this Notch3 gene, and that 160 is probably at least 5 years old. It wouldn't surprise me if there are many more than this by now. There are cases that do not have a Notch3 mutation, but clinically, radiologically, meaning on brain MRI, and if we look at the skin biopsy, they have the same findings as a genetic patient with CADASIL, but they don't have a Notch3 mutation, and they don't have a family history. So the disease can occur sporadically or spontaneously without a mutation, and we're not sure why that is. Now, many diseases are both sporadic and genetic, but usually the sporadic forms are much more common. For example, Alzheimer's disease is usually a spontaneous or sporadic disease. Less than 2% of Alzheimer's disease is actually caused by an autosomal dominant mutation. There are at least 3 genes that can cause it that lead to the same outcome as Alzheimer's disease. But the genetic forms of Alzheimer's disease are much less common, whereas in CADASIL, it's the reverse. The genetic forms are much more common, and the sporadic cases, to our knowledge, are less common. So the CADASIL is primarily due to mutations or errors in the gene called Notch3. This gene is on chromosome 19. We have 23 pairs of chromosomes. 19 is one of the pairs. Each chromosome has 2 strands. One strand we inherited from our mother. The other strand we inherited from our biological father. And chromosome 19 happens to be where the Notch3 gene is, and that's where the mutation is. So this gene provides instructions for making a protein that spans the cell wall or the membrane, particularly of smooth muscle cells in blood vessels. And that's depicted, this protein, the Notch3, is shown in the orange color on the picture on the right. And you can see this protein is actually a receptor. A receptor is a protein that binds other proteins. And the top part of the protein here is the receptor, and other proteins called ligands bind to this. Part of the Notch3 protein is in the cell membrane. It's in the cell wall. So the top part of the figure is outside the cell wall, and the lower part of the figure is inside the cell. And deep below this picture would be the nucleus, where the genetic material, the DNA, resides. And what happens is, when a protein ligand binds to the outside of the extracellular domain of this Notch receptor, the intracellular, the inner part of the protein, which here is called the Notch3 intracellular domain, or NICD. This part of the protein actually breaks off and it goes into the cell. And I depict that here in this next figure. So the same Notch3 gene shown here, spanning the cell membrane. And when a ligand binds to it, that NICD breaks off and it goes into the cell, and it actually gets into the nucleus. And it actually acts on other genes and causes other genes to become active. And what do these other genes do? Well, these other genes shown in pink and in white, they all seem to interact, be affected by this NICD domain. And ultimately what happens is that these genes play a role in the function and survival of smooth muscle cells of the blood vessels, the small blood vessels in the brain. And so Notch3 is essential for maintenance of small blood vessels in the brain. And if there's a mutation, this protein doesn't work right, and the Notch3 doesn't get all of the nutrients, it doesn't work quite right, and eventually it will lead to disease. And the disease, when we look under the microscope, after somebody dies from this disease, we look under the microscope and we look at the blood vessels in the brain, and those are shown on the left side of the figure. And what you're seeing, you're looking at a cross section, thinking of cutting a pipe in half. And that white part in the center is the hole or the lumen of a pipe, or in this case, a blood vessel. And all that red material that you're seeing, that dark red material and the pinkish material, that's too much. That shouldn't be there. There should be very little of that here. And we call this granular osmophilic material, or GOM. And it's one of the ways that we can make the diagnosis. And we can actually see this not only in the brain, we see this in the skin, but for some reason it seems to only be the brain that's affected. We can also use other stains shown in the 2 pictures below to help identify the material that's in the blood vessel wall. So you can see that the lumen itself is clear. We don't see plaques that we would typically see in a patient who has arteriosclerotic disease from having a poor diet, from having diabetes, etc. The vessel wall is clear, it's just very small. And on the right are really high magnification, actually at the using electron beam microscope. So these are electron micrographs on the right, and on the top is low power, so low magnification. And what we're seeing is the smooth muscle cells are shown in S here, and here is some in the vessel wall, and the arrows are pointing to this material that shouldn't be there, this material that's building up because of the Notch3 mutation. It's not being processed right. And then below is a high magnification image of that, and S is the actual outline of the smooth muscle wall, and then the arrows are pointing to this granular osmophilic material, or GOM, that shouldn't be there. And this is what's causing thickening of the vessel walls and improper function of the smooth vessels of the wall. So the walls become too thick. So that's so we can make a diagnosis of CADASIL by doing a biopsy of the brain, a biopsy of the brain white matter, or sometimes of the skin, and we can do an electron micrograph of that to make the diagnosis. We can also make a diagnosis in most cases doing genetic testing, and as that's much easier, that's sort of the more common way of making the diagnosis, particularly for the genetic forms of the disease. In the rare sporadic or less common sporadic forms, there is no mutation, so we depend upon these pathological findings in the skin or the brain to make the diagnosis. So I've mentioned several times the term autosomal dominant, and here I'll try to describe that, autosomal dominant family history. And what we're showing here are 3 different generations of the family. Level 1, Roman numeral 1 on the left, you can see it, in this case, it would be the grandparents of the 3rd generation. So the square is a male and the circles are all female. And blue indicate people who carry the Notch3 mutation. And you can see in this case that the father has a mutation, and then he has his biological children are shown by the lines. So he has 4 children, and unfortunately 3 of them have inherited the bad gene, or the Notch3 with the mutation. So every person has 2 copies of every gene, and in this case the gene is on chromosome 19. So there's a mutation on the father's chromosome 19, one of the 2 copies of the father's chromosome 19. He got 1 copy from his mother and 1 from his father. In this figure, we don't know which, whether it was his mother or father that gave him the bad copy. In this case, each child could get a good copy or a bad copy of the gene from the father. They get 1 copy of Notch3 from the father and 1 copy of Notch3 from the mother, just like every other gene in the body. They get 1 copy from each parent. So when something's autosomal, it's passed down by it's not passed down by sex chromosomes. The sex chromosome is the 23rd chromosome. Chromosomes 1 through 22 are the autosomes, or the not sex chromosome, in this case, chromosome 19. So the disease can occur in any sex. It can occur in men or women. If it were on the sex chromosome, the 23rd chromosome, then there would be a gender difference. And in CADASIL, there isn't a gender difference in terms of one's risk for getting it. And it's dominant, meaning that you have 2 copies of every gene. For dominant diseases, only 1 copy has to be bad or have a mutation in order for you to get the disease. In a recessive disease, such as some blood disorders like sickle cell anemia, you need to have both copies to be mutated, meaning the copy you got from your mother and the copy you got from your biological father, both would have to be mutated. That would be a recessive disorder. This is dominant. You only need to have 1 copy. And that means that each child has a 50% risk of inheriting the disease from a parent who has the mutation. In this case, 3 of 4 children developed, unfortunately inherited the mutation, but it's sort of, it's a crapshoot. It's like being at the crap table where each person has 50%. If you have a family of 10 children, statistically, you would expect about half of them to have it, but each person is 50%. If 9 children in the family have the mutation, the 10th child, their risk is still 50%. Each child is independent of every other child. They each have a 50% risk. And if you look at all CADASIL, it's about 50%. But in one particular family, you might see a little bit different. So in the 3rd generation here, you see that this the father's son has 2 children, and unfortunately one of them, the daughter, received the bad gene, the notch mutation. The other son did not inherit the mutation, so there were 50% who didn't get it. This daughter got it, but hasn't yet had children, so hasn't passed it on. This daughter, again, did not get the mutation, and has no children yet. And in this case, this daughter did get it, she married, and 2 of her 4 children have gotten the mutation. What you'd expect just based on statistics. Each child has a 50% risk. So that's a pretty complicated concept. I hope that was somewhat clear. So let's talk about the clinical symptoms we see in CADASIL. The 4 main symptoms are migraine, which is a form of headache, and it's not just any headache. Migraine defines a certain type of headache, and usually patients often have pain. The pain often has certain locations. It can be very intense pain. The pain usually lasts for hours. There often are other symptoms associated with the headache. Bright lights can bother people. Sounds can bother people. Patients often want to go to a quiet, dark place. And migraines are very common. A large percentage of the population, or a large minority of the population, suffers from migraines. So it's a pretty common disorder, and it's even more common in CADASIL. Aura. An aura are symptoms that occur before the onset of the migraine. And these, I'll discuss a little bit more what those are later, but there are other symptoms that precede the migraine that tell you a migraine is about to happen. And then the other symptoms are strokes. Ischemia is a term that we use to say that there's a decrease of blood supply to a certain region of the brain or a certain region of the body. If there's enough ischemia, enough blockage of blood flow in a blood vessel, it can lead to a stroke, meaning that the tissue is injured and can die. And we already talked about subcortical, meaning that's the deep white matter of the brain, and the deep structures in the brain are affected, not typically the surface of the brain, which is the cortical area. And then mood and psychiatric symptoms are relatively common. Apathy, so I'll talk more about these. And then cognitive decline, and eventually the cognitive decline becomes dementia. Dementia is a term that means somebody has a decline in cognitive function, and cognitive function means memory, executive function, which is the ability to plan, organize, multitask, maybe something that an executive would have to do. Visual spatial function, how the brain processes vision and imaging. Language, those are all cognitive domains. If you have impairment in cognitive domains to the point where you're no longer able to function the way you used to be able to function, that is by definition dementia. Dementia is an umbrella term. The most common dementia would be Alzheimer's disease. The next common would be vascular dementia. And CADASIL is one cause of vascular dementia. Other causes would be Parkinson's disease can cause dementia, Huntington's disease, sometimes metabolic disorders. So dementia is an umbrella term And there are many different types of dementia. CADASIL is one type. So we talked about migraine being one of the 4 most prominent symptoms in CADASIL. These migraines occur in about 20 to 40% of patients. So the majority of patients with CADASIL might not have a migraine. Now this incidence, this occurrence of migraine is 5 times more common than the general population. And it's particularly migraine with aura. And aura often, they're visual or sensory symptoms. Often they can last for minutes, 20 to 30 minutes, sometimes less. And the visual symptoms might be wavy lines in the vision, or even partial loss of vision or darkening of the vision. Sensory symptoms might be numbness or tingling in one part of the body, an arm, a leg, half of the body. And those are what we call auras. And the migraines in CADASIL usually are migraine with aura. Now, migraines without aura are probably just as common in CADASIL as they are in the general population. But migraine without aura, that is. But migraine with aura, much more common in CADASIL than the general population. These migraines, on average, occur around age 30, but there's a wide range, age 6 to 48, maybe even later. Interestingly, in women, it's a little bit earlier, I believe, and later in men. And I think it's about 26 for women is the average age, and for men it's 36. Now, about 50% of people who have the migraines have atypical features. So they actually have weakness. They can have prolonged symptoms lasting more than just 20 to 30 minutes. They can have confusion. The frequency of how often these migraines occur varies greatly from patient to patient, even within families. And even within families, some patients have migraines with aura and some do not. So psychiatric symptoms. Well, the literature says 20% of patients have mood affected. In my own experience, it's actually probably much higher than this. I just think that often it's not asked about. It's very important to inquire about it. Patients often won't volunteer that they're depressed. Unless you ask about depression, you may not identify it. Apathy or decreased motivation occurs in about 40% of patients. Mania is very rare. So this is when people have become very excited, have rapid thoughts, manic disorder. And then psychoses, visual hallucinations, fixed false belief or delusions can sometimes occur, very rarely. And then strokes. More than 2/3 of patients have strokes. And a stroke is can be defined as several ways, but a stroke is where you have blockage of blood flow to the brain. And in CADASIL, it's because of the small vessels in the brain. They get narrowed and not enough blood gets to a certain part of the brain. That causes ischemia or loss of oxygen and other nutrients that blood supplies to that area of the brain. The nerve cells supplied by that blood vessel become injured and can even die. If the symptoms last more than, if somebody has symptoms and they last more than 24 hours, we call that a stroke. If they have symptoms that last less than 24 hours, we use the term TIA, which stands for transient, meaning temporary, ischemic attack. So there's symptoms that last less than 24 hours. And these strokes can occur in many different ways in the brain. Lacunar means they can cause small, tiny holes in the brain. Sometimes we can see these in MRI. They tend to be very localized. As we mentioned, they're subcortical. They're usually not on the surface of the brain. And as we mentioned, they can be often in the white matter, or they can be in the deep gray matter. So as I mentioned earlier, the neurons, the nerve cells in the brain tend to be on the surface of the brain, and those tend not to be as affected in CADASIL. Or there are collections of nerve cells called ganglia or nuclei in the center of the brain, and those can be affected just as much as the white matter in CADASIL. Why those areas are affected more than the surface of the brain is not well understood to my knowledge. And you can also get involvement of the brainstem. So the brainstem is the part of the brain that's sort of in the back of the head that connects from the spinal cord to the brain. And the spinal cord sometimes can be involved as well, but less commonly. So the dementia occurs in about up to 80% of patients. In my experience, it's probably even higher, but then again, I work in a dementia clinic, so perhaps there's a bias there. It's the strokes that cause the cognitive decline, leading eventually to dementia in most cases. And the dementia is what we call frontal lobe dementia or a subcortical dementia. And what that means is that it's a different type of dementia than we see in Alzheimer's disease that particularly affects an area of the brain called the hippocampus, which is an area of gray matter. And in Alzheimer's disease, we usually have trouble recording memories. We just can't record memory or encode memory. But in a frontal dementia or subcortical dementia, it's a different problem. Patients have trouble organizing, trouble planning, trouble multitasking. They have trouble with processing information that's given to them. Their thinking is a little bit slowed. Now, sometimes these can manifest as a memory problem. Because they're slow thinking, you tell a patient who has CADASIL information and they may not be able to process it quickly enough. And so that can look like a memory problem, but when we do more sophisticated cognitive testing in our clinic, we can actually determine, is the memory problem due to frontal executive dysfunction, or is it due to a problem in encoding the memory, such as we might see in Alzheimer's disease. And in one study of 42 patients who had some symptoms of CADASIL, every patient from all patients aged 35 to 50 had deficits on cognitive testing. So this is why I think more than 80% of patients actually have cognitive dysfunction. Unless you actually look for and test for it, it may not be obvious. Now, in some cases, it may not affect somebody's function because maybe whatever they do doesn't require a lot of frontal executive dysfunction, or maybe they're able to compensate for the deficits, but we can often detect those deficits on cognitive testing. And eventually, as the disease progresses, more than frontal executive dysfunction occurs. We do get problems with memory. We do get problems with other cognitive domains. So visual spatial domains can be affected. Problems with language can occur. But those are usually later in the disease. We don't always know when the onset of cognitive decline begins. As this previous study mentioned, when they looked at patients as young as 35, they found it in almost all patients in this small study. But we don't know really what age it begins. As I'll say later, the MRI changes can be seen usually starting around age 25. So it might be expected that we might even see cognitive changes back then if we really did detailed testing, although it may not be obvious clinically. Patients might not have symptoms. So 4 out of 5 patients who are over the age of 65 with CADASIL will have dementia, and about 10 to 20% will develop dementia without clinical strokes. And what that means is without having obvious clinical symptoms of stroke. So they may not notice that they had a stroke, even though on MRI we can detect it. So for 1/3 of patients, the first symptoms are migraines, and that means for 2/3, it's not migraine, it's something else. Usually the migraines begin in one's 30s, but as early can occur as even as early as 10 years old or younger. The most severe symptoms usually are strokes, and usually these occur in one's 30s to 50s, and the peak age is usually age 40 to 50. And usually once these start, usually patients will live another 10 to 20 years. But most patients can live into their 60s and a relatively normal lifespan, though the latter years usually patients are impaired to a mild extent, eventually to a larger extent as the disease progresses. So we've talked about brain imaging or MRI in CADASIL. And I already mentioned that the MRI can detect changes beginning in one's mid-20s before most other symptoms develop. And I'm going to show you several images because I think pictures can sometimes be much easier than looking at text. And because the vessels in the brain are affected, they become narrow. One test that we try to avoid, and the vessels are more friable, or they're more easily broken because of the deposition of the abnormal material that I showed you occurs pathologically. We tend to avoid procedures in which we have to put wires up into the brain to look at blood vessels. So there's a technique called cerebral angiography, where if a patient's having stroke-like symptoms, we actually might put a catheter or tubing into a vein, into an artery in the leg, and actually put it through the vessels all the way through the body and go up into the brain. And then we put a contrast agent into that tubing, and we put it in the brain, and then take a picture of the brain, and we can really see the outline of the vessels very well. We do that in patients who have strokes, and we're trying to see why they might be having a stroke, to detect exactly which blood vessels might be involved, to see if we can repair it. Well, that technique can cause damage to patients with CADASIL and might lead to even a worse outcome. So we usually recommend avoiding cerebral angiography when it's invasive in patients with CADASIL. Here I'm showing you brain MRIs on patients with CADASIL. And this is one patient, and there are three different, each row is a different type of MRI technique. And what we're showing is in the top part of each image is the front of the brain. The back part is the back of the brain. And we're looking at on top of the brain. So we're looking a bird's eye view, looking down on top of the brain. And here, these dark structures, and with the white behind them, these are the eyes. Here is the muscle of the eye. And the brain starts back here. These are what we call the temporal lobes. This is the cerebellum, and we're seeing some other back part of the cerebellum here as well. The arrows point to these. There shouldn't be these dark holes within the tissue. They shouldn't be there. This shouldn't be there. This shouldn't be there. These. Here, this is a normal hole. This is just where the spinal fluid goes. But these smaller holes are abnormal and shouldn't be there. Now, as we're starting down low in the brain, and as we move to the right, we're moving up to the top of the brain. And here, these are the deep nuclei. These are gray matter structures. These are deep nuclei too. And you can see there are these holes here. These little holes should not be there. And this is the white matter here. On this sequence, it doesn't look white. But if we actually took the brain out and looked at it with our eyes, this area actually would be white. And you can see here, it's a little darker than it is here. This is normal. This is abnormal. And the arrows are pointing to all these abnormal areas. Here are some kind of holes that occur in the white matter as we move up. Now, this is a different sequence. The second row is called a flare sequence. And most abnormalities in the that we see in the brain will appear bright white on this sequence. So most of the things that are bright white within the brain tissue itself are abnormal and shouldn't be there. And here, bright white, this is all abnormal. These areas here are all abnormal. And you can see there's a little black hole there. These are called lacunes. Lacune just means a little valley or a sort of an outpouching. And again, here, all the white matter in the brain, and these are little holes. We call these cavities, or like you get a cavity in a tooth, except here it's a cavity in the white matter of the brain. Now, the last sequence, the last row, again, the same patient. In this sequence, I call this a hemosiderin. Hemosiderin is a term that we use for the breakdown products of blood. So if there's been blood in the brain that leaks from blood vessels, eventually we'll see iron deposition of iron, which is a component of blood, and it will deposit. And areas that have blood in them, old dried blood, will be dark on this sequence. Now, this is just artifact here, but what isn't artifact are where I'm pointing to now and where the arrows are pointing to. So this patient has small bleeds in the brain, tiny, tiny bleeds. Now, just so you know, this image actually magnifies the size of these bleeds, probably 5 or 10 folds bigger than normal. So they're actually much smaller than depicted here on this image. So they're very tiny, but this image magnifies them. So this patient has these bleeds, and they're often in these deep nuclei, these deep gray matter structures, and sometimes they're in the white matter. And so this patient would be one of the 30% of patients, 20 to 30% or so, maybe a third of patients with CADASIL who have these bleeds. So that means that the majority of patients we don't find with CADASIL, we don't find these bleeds. So here's one of my patients. This was a 55-year-old woman who had had CADASIL since about the age of 35. Note, there are 3 classic findings on this MRI. Again, this is the front of the brain. We're doing a bird's eye view. This is the back of the brain, the back of the skull. By the way, this white structure is actually the skull, and this white behind the eye is fat. Behind the eyeball is a lot of fat that cushions the eye, and that's all normal. But looking at the brain itself, this white should not be here. And in this case, these are called the temporal lobes. These are lobes, as you can tell, right behind the eye. And there's white matter here. There are very few diseases that can cause white matter in this pattern. And one of the most common would be CADASIL. Now, another common area to see these white matter changes in CADASIL is here. So this is a slice up, and this kind of would be, the ears might be right over here on the side. So this kind of just in front of and inside from the ear, there's an area called the insula, and this is an area that often is affected in CADASIL. And then as we go a few images up, here we're looking at the top of the brain, and here you can see the white matter. Now, you can see that the white matter here is what we call confluent. So you're not seeing all these small little, what we call punctate or small little ditzels here. And the reason why it's more confluent here is because there's so many of these white matter ditzels, they've become confluent, they've coalesced. And so if we looked at maybe this patient's MRI 20 years ago, we might have just seen a few little ditzels like this, little dots here. But over time, these dots, more and more come of them, and we can no longer see anything but white because there have been so many small silent, so many strokes. And this dashed line is pointing to a cavitary lesion or a hole in the white matter that can occur from the strokes. Now, I'm not showing you the image looking for blood products, but as with the majority of patients with CADASIL, this patient did not have those microhemorrhages. Here's another patient published in the literature, and in this case, this is an image called a T2 image. It's very similar to the flare, except here we're looking, the spinal fluid is white on this image, so this is normal. But within the tissue, and this spinal fluid here is all normal too. So the spinal fluid that was dark on the previous image is white on this image, and that's normal. But what's not normal is within the brain tissue itself, not within the fluid surrounding inside or surrounding the brain. Within the tissue, we're seeing all this white that shouldn't be there. And this is consistent with CADASIL. In this case, this is actually a large stroke. There's breakdown of the tissue. And in this case, the patient had a larger stroke, which is a little less common in CADASIL. But here we're seeing some small little lacunes also. And here, this is an image of the brain, a flare image. So in this case, the white is all abnormal. The dark here is the spinal fluid, and that's normal. But in this image, we're looking at the brain almost like slicing a slice of a loaf of bread, and we're looking straight on. The eyes would be in front of us, the back of the brain would be behind the screen. And here the temporal lobes that I showed you before, so the eyes would be right in front of this, and you can see the white matter in these temporal lobes. This area of involvement is very classic for CADASIL. And it's usually on both sides of the brain, meaning it's symmetric. It's usually throughout the brain, meaning it's diffuse. And here are, again, the specific MRI features. Again, we're seeing the front of the brain. Here's actually the nasal cavity, the top of the nasal cavity. These are the eyes. These are the temporal lobes right behind the eyes. And these arrows are pointing to enlarged vascular spaces that we see in CADASIL on this image. And on this image that's just a little bit higher up, we're going up a few slices. We see there's white matter as well. And there might even be some breakdown of blood actually on this image as well. This patient might have some bleeds. And so this pattern of anterior temporal lobe involvement with white matter, these bright white areas, is seen in about 2/3 of patients. So about 2/3 of patients don't have anterior temporal lobe involvement. And that can make the diagnosis a little harder because then we're thinking of many causes of cerebrovascular disease, such as high blood pressure, hypertension, high lipids, diabetes, or other causes of white matter disease, of which there are dozens and dozens. And I'll talk about that in a little bit. Again, these are two of my, two patients, a classic MRI again, an area very involved beneath this area of the brain called the insula. And here we're looking higher up in the brain, and then the anterior temporal lobes involved. This is a patient of mine down below who actually had a non-classic, late onset form of CADASIL. And you can see the anterior temporal lobes, the white matter is actually pretty normal. There is no problem in the anterior temporal lobes. Only when we look higher in the brain do we see that there are lots of strokes and problems in the white matter. So here's a description of a patient of mine. She was a 74-year-old woman who was right-handed. She had diet-controlled diabetes that was well controlled, but had difficulties with balance that was unexplained for several years. She had headaches that were on a pain level, 10 being the worst pain one can imagine. Hers were 5 to 6 out of 10. 0 out of 10 would be no pain. They occurred daily. They lasted for hours. They were dull ache. There was no photo or phonophobia, which means lights didn't bother her, sounds didn't bother her. So not a classic migraine like we would see in most CADASIL cases. She does have flashing lights, which is some of what we call the aura or visual symptoms. And the MRI, as I'll show in the next slide, did show white matter disease. It's abnormally bright. But she reported no memory, visual, language, cognitive issues, no other motor problems, and some balance. And she still was functioning well, living independently. On a brief cognitive test called the mini mental status exam, which is a 30-point test that takes about 10 minutes to do in the doctor's office, she got 29 out of 30, which is normal. 28 to 30 is normal on this test. But this test does not test frontal executive function. It doesn't test the cognitive domains that are affected in Alzheimer's disease. It's much better for Alzheimer's disease detection than it is for CADASIL. She had some mild problem with remembering a list of words that we gave her. And she had a lot of problem on what we call frontal executive dysfunction. When we give her tests of how quickly she can process information, organizing, planning, multitasking tasks, that's where she had trouble. Even though she was still functioning well at home, we could detect problems. And her MRI, this is her MRI. You can see all these bright white areas should not be here. She has problems in the deep nuclei. She has problems in the white matter. But what she doesn't have is she doesn't have much anterior temporal lobe involvement, a little bit here, especially for somebody who is as old as she is at 74 with CADASIL. It's a little bit unusual, and she doesn't have a lot of cavitary lesions. This is an image of the brain looking at it from the side. If you're looking at it from the ear, so the front of the brain would be the front of the head, the nose is to the left. This is the mouth here, the nose, and here's the back of the brain. Here's the spinal cord, right? And here is the spinal cord. So you can see the white matter disease as well. So in this case, because the MRI was atypical, usually when we're testing for CADASIL, we do genetic sequencing where we look exactly for the Notch3 gene. So if we really think CADASIL, it's much cheaper, it's much quicker to just look for mutations in the CADASIL gene. In this case, we didn't do that because I had many other diagnoses in my list. I gave the family a list of 10 other conditions I thought that could cause this. And we realized that if we went through that list one at a time, it could be tens of thousands of dollars. And at that time, we could actually sequence her entire genome essentially for a few $1,000s. And that's what we did. It turns out that it was CADASIL. And the CADASIL test probably would have been $500 to $1,000. This testing made for whole exome sequencing might have been $2,500 to $3,000, some of which was covered by insurance. So it was an more expensive way than we would have liked to have diagnosed CADASIL, but because she didn't present classically and we weren't sure what we were going to find, we did whole exome sequencing. For atypical forms of CADASIL, that's often how we make the diagnosis. So this slide is meant to summarize some of the symptoms and signs that we see in CADASIL over time. So on the lower part of the axis of the graph, or the x-axis, we see age in years going from 10 up to a person's 80s. And then the colored boxes show typically when different symptoms or signs begin in CADASIL. So the leftmost is migraine with aura, often beginning, let's say in this case, average age around 15, but there's, as I've told you, there's a wide range. And then MRI abnormalities. Usually we see these by somebody's 20s. To be honest, we often don't do MRIs before this age because patients don't have symptoms. So it's possible that they are occurring earlier. We just often haven't looked, and that's why there's a dotted box here. And then stroke, often occurring between one's ages 30s to 50s, as I mentioned. And then mood disturbances. Now this, I think, is variable. We can often see these even earlier. Apathy, so loss of interest in many activities. Motor disability, so problems swallowing, problems speaking, problems using one's limbs, problems with gait, problems with urinary control. These are all motor function. So that tends to occur usually after one's 50s. And then cognitive function usually probably begins sometime in one's 30s, although may not become obvious to the patient or to colleagues or family until later in life, but we can find it on testing early. And then dementia doesn't occur usually until later in the disease. And patients often can live a relatively normal lifespan, probably maybe dying a little bit earlier than average, particularly as over the past few decades, average lifespan has increased so much in healthy people. So how do we make the diagnosis? Well, the clinical history of the patient, key features as I've gone through, finding a family medical history that's consistent, that autosomal dominant family tree that's consistent with CADASIL. Do we see persons in multiple generations with symptoms suggestive of CADASIL, migraine, strokes, motor problems, cognitive dysfunction. Now, realize again that not everybody will present with the same symptoms. So some family members may not have migraines, others will. Some might not have obvious strokes, some might not have obvious dementia. The brain MRI, I've shown you many, is the MRI suggestive? I've shown you many figures for that. And then usually the next step is genetic counseling. If we're really considering it, we usually send patients to a genetic counselor, and that we use a protocol that we use for another neurogenetic disease called Huntington's disease. And it's a protocol established by the medical community that establishes guidelines for how we do testing. We don't do genetic testing without counseling. We think that's unethical because we want patients to make sure that they have full knowledge of what the implications are of finding out a test. if we give a test result, has the patient thought about how they're going to tell other family members? How are they going to tell their siblings, their children, their cousins, their aunts and uncles, their nieces and nephews? What's the plan? Have they thought about what they're going to do? How will knowing, getting that result affect them? We ask about depression, suicidal thoughts. If they got the genetic result, might they do harm to themselves or others? These are all part of the counseling protocol that's usually done by a genetic counselor or a medical personnel who's trained in such things. If we feel that there is depression, we think depression might be worsened by finding out their genetic result, then we intervene, making sure that the patient's depression is being managed before releasing the results. Usually we want to know, do they have a support person, a family member, a loved one who can be there for them, a support person, a therapist, somebody. If the family mutation is already known, so we know the specific Notch3 mutation, so other family members have already had genetic testing that has been positive, then we can test much more specifically for the same mutation, and that test is quicker and less expensive. If that hasn't happened, we have to actually sequence the whole Notch3 gene, and it's a little bit more expensive. If this mutation is negative, or for whatever reason, the genetic testing is not available, and it may not be available in all countries, then a skin biopsy is done looking for the pathological changes in the blood vessels that I've already discussed. We see changes in the blood vessel walls, and we can do staining on those with antibodies looking for particular buildup of the abnormal protein that occurs, the granular osmophilic material. And so sometimes we'll do a skin biopsy, and this is quite important in patients who have sporadic disease in whom there is no mutation. Now, this slide is a diagnostic decision tree. It's meant to tell you it's complicated. You're not meant to look at this slide and understand it, but I'll briefly take you through part of it. So we might be suspicious of somebody who has CADASIL, maybe because of the family history. And in the top left, we see somebody has white matter abnormalities on the brain. And if they have some cognitive impairment, then this takes us down another decision tree. If they have migraine, stroke, mood disturbances, apathy, in red would take us down a workup for CADASIL. Does the MRI show particular features that are suggestive of CADASIL? Is there an autosomal dominant family history, meaning both genders are affected? It doesn't tend to skip a generation. Every generation is affected. And then if so, then we might do genetic testing, genetic counseling, genetic testing, looking for the Notch3 mutation. If we find it, it's CADASIL. If we don't find it, but we're still suspicious, we might do a skin biopsy. And then if there were other features present, if this was all negative or there are other features, we might look for many of the other diseases that are shown in this slide that I will not discuss with you today. But this is from a chapter discussing CADASIL in a book that I've published with several colleagues, including other CADASIL experts that I'll talk about at the end. How do we manage CADASIL? Well, let's talk about each symptom. So the migraines, one thing we want to do is we want to, in some cases, if a patient is having a lot of migraines with auras, we might, a lot meaning several times a month, maybe more than once a week, maybe twice a week, we might want to put them on a medicine that prevents them from happening. And the other term for that is called prophylaxis. We want to prevent a migraine, just like we use the term prophylaxis for pregnancy. We want a prophylactic is you want to prevent pregnancy, so you use a prophylactic. In this case, the term prophylactic is referring to prevention of a migraine. In this case, migraine with aura. So there are many medicines we use to treat migraines with aura, anti-epileptics, beta blockers that we use as preventative. Acetazolamide is a diuretic. It's a medicine in a class called a diuretic. And a lot of anecdotal evidence suggests that it's very helpful in preventing migraines in CADASIL. And to my knowledge, there's been no formal study, but just an abundance of observational data showing that this drug seems to really be helpful. This is a drug that's used not only as a diuretic for people with high blood pressure, it's also used for altitude sickness, for prevention and treatment of altitude sickness. Now, acute or abortive treatment, what this refers to is acute, meaning you're having a bad migraine now. How do you treat it? How do you make it, lessen it, and make the migraine go away? How do you abort the migraine? And we tend to avoid medications that we use in people who don't have CADASIL but have migraines. And those are drugs such as ergot, alkaloids, or triptans. These are drugs that actually affect the blood vessels. And the reason we tend not to use these is because there's a theoretically we are concerned that it could worsen the blood vessels. It might help the migraine, but might worsen strokes. The data on this, I don't think is that strong. It's more empirical, and I think most CADASIL experts tend to avoid these. But I know some of my colleagues do use these in patients with CADASIL. I do not personally. And then instead, we tend to prefer conventional pain medicine, acetaminophen, the non-steroidal anti-inflammatories, naproxen, ibuprofen, etc. But we tend to try to only use these in people who have infrequent migraines, a few times a month, a few times a year. If they're very common, then we might try to put a patient on a preventative treatment. What about the strokes? Well, in general, it's similar treatment to those patients who have cerebrovascular disease from non-cardioembolic sources. So cardio meaning heart, embolic, and embolism is a blood clot that forms in the heart. So and it leaves the heart by the blood vessels and it goes to the body, either to the heart or to the body. That's an embolism, and embolisms can go to the blood vessels in the limbs, to the lungs. It can be a pulmonary embolism, or it could go to the brain. And if it goes to the brain, it can cause a stroke. So for patients who have cardioembolic causes of stroke, we tend to use blood thinners. We tend to use anticoagulants. And those are usually avoided in patients who have CADASIL because we think theoretically that might cause worsening of the bleeding in the brain. It can exacerbate the disease. Instead, we tend to use drugs that prevent the clots from forming in the first place, antiplatelet drugs. So aspirin would be a typical drug.
You're the only participant in this conference. Press any key to continue this conference. The conference will now continue.
So other drugs that can occur, other treatments that can occur that we would use would be the antiplatelets, but we tend to avoid any blood thinners. If there's high blood pressure or high cholesterol, we tend to treat those very aggressively. In general, we think what's good for the heart is good for the brain. So a heart-healthy diet, an active cardiovascular lifestyle. I usually recommend for my patients gradually working up to a cardiovascular exercise program of at least 30 minutes, at least 3 times a week, ideally 5 days a week, and whatever exercise they like. Rapid walking. It should be exercise to the point that you're breathing heavily enough that a casual conversation would not be comfortable. It could be an exercise bike. It could be swimming, aerobics, whatever works for you, whatever you're willing to do. And again, we generally avoid clot-busting agents such as intravenous tissue plasminogen activator or TPA or streptokinase, and we avoid blood thinners such as heparin or warfarin. And this is because there's a theoretical risk of bleeding. Now, some of my colleagues who manage patients with CADASIL, if a patient doesn't have known bleeds on their brain imaging, so then they might actually be willing to use these agents. And we really don't have the answer to whether or not this is safe or dangerous. But this is something that is a decision made by the individual physician, and sometimes with in talking with the patient or the patient's loved ones. I tend to not give these agents because the risk is too high, but every patient I treat individually. Most of our patients, we recommend wearing a medical alert or a similar type of bracelet, necklace, or something in their wallet, warning, saying that they have CADASIL, having a number to call, a website to look at, one of the CADASIL organization websites, and a family member or loved one to call in case of an emergency. And then we tend to avoid invasive angiography due to the blood vessels being friable. How do we manage the cognitive impairment or dementia in CADASIL? Well, there's only been one treatment trial looking at the cognitive impairment in CADASIL, and this is a trial that I was very fortunate to be a site investigator for. But it was a international trial, multiple sites around the United States and elsewhere. And we used the drug used for Alzheimer's disease, a drug called donepezil. It was one of the first drugs approved for the treatment of the cognitive symptoms of Alzheimer's disease. In this trial, 168 patients with CADASIL who had some degree of cognitive impairment were enrolled. They were treated for 18 weeks. So this isn't a very long trial. And unfortunately, the primary outcome of the trial was what we call a cognitive subscale of vascular Alzheimer's disease assessment scale. And so in retrospect, it probably wasn't the perfect outcome. And we didn't find benefit in that. But we did find improvement in several aspects of executive function. And because these weren't the primary outcome, technically we had to call the trial, the drug unsuccessful. But because we saw success in some of the secondary outcomes, I think many of us interpret this data as being suggestive that CADASIL might be helpful in some patients. I offer it to all of my patients with CADASIL who have cognitive complaints. And I can't give a precise number, but certainly a large minority of them, either they feel or their loved ones feel that there is some improvement. If they don't feel that there's improvement, I usually would stop the drug after a few months. Sometimes when they stop the drug, however, they notice that there's a decline. And if that happens, I quickly get them back on the drug. Sometimes we can use tests in clinic to see if they actually improve, but I tend to rather take input from the patient and family. Do they feel better on the drug? Are they sharper? Are they more alert? Are they more conversant? Whatever is better, I'm looking for any kind of improvement. And it may not be on a pen and paper testing like the mini mental status exam or the MMSE that I see improvement. So I think what this trial told us is that future trials will need to assess executive function measures such as processing speed. How quickly can you do a task that has you alternate between different tasks? That's something that we call processing speed. How do we manage other aspects of CADASIL? Well, for depression and mood, it's pretty much standard of care. Therapy, make sure my patients have a therapist, a psychiatrist, or a counselor. Sometimes we use medicines. Every doctor might be more or less comfortable with using medicines for depression. The SSRIs are common drugs that are used, but there are many drugs we use for depression. I try to recommend stress reduction. If the patient has a stressful life, I try to work with them or their therapist to try to reduce that stress. I already talked about exercise. That can be good for not only cognitive function, but also for mood and depression. We think it also might be somewhat protective to the brain. So that's why I put all of my patients into an active cardiovascular exercise program. I want to get good blood flow to the brain. And then there's a technique called mindfulness-based stress reduction, or MBSR. It's offered in many places. You can just look on the websites, search engines for it. And it's a technique that can help patients focus. I use this for cognitive dysfunction in not only CADASIL, but many disorders, for mood disorders, for apathy. It can be quite helpful. It's basically a form of meditation. The class is it's usually an 8-week class where you go to class for 2 hours, but you have daily homework that you have to do for an hour, where you have to practice what they teach you. And then you try to incorporate it into your life after the 8-week class ends. Other things, if patients are having motor problems, we give physical therapy. If they're having problems in their home, we might give occupational therapy. We might provide psychological counseling therapy. I often have my social worker work with them. Nursing care, if they're in the later stages of disease and have a lot of dysfunction. Genetic counseling can be important, of course, for the patient and the family. This starts at the time before we even do genetic testing. How can you help? Well, I say get involved in research. Because it's a relatively rare disease, there's not a lot of funding for this disease, and there's not a lot of research in it, but there are researchers around the country who are working on this disease. And there's a study called the MARC-VCID. This is a study that the VCID stands for Vascular Contributions to Cognitive Impairment and Dementia. Not a great acronym, but it's what they came up with. And this is a consortium of United States academic medical centers. They're trying to identify and validate biomarkers. And biomarkers are markers of disease other than the disease itself. So it might be a protein that we find in the spinal fluid. It might be a change on MRI. It might be a cognitive test. To validate biomarkers that occur in small vessel diseases of the brain, like cerebrovascular disease, one type of which is CADASIL, and that produce or cause cognitive impairment or dementia. And the goal of this is to deliver high quality biomarkers, so high quality outcomes that can be used for clinical trials, so that we might actually be able to develop trials more quickly, more efficiently, more cost effectively, and get treatments to patients quicker. Here's the website for this study, marcvcid.partners.org. This is the main website, and there are several sites on this website that are listed on this website, including UCSF. And you can look on the UCSF Memory and Aging Center website as well, memory.ucsf.edu, to find out, and you can look under studies or clinical trials to find out more about this study. Importantly, through the advocacy of different organizations such as CureCADASIL and others, there's a new diagnostic code for CADASIL. For decades, we've never had a diagnostic code for CADASIL. We had to use diagnostic codes that refer to more larger terms, umbrella terms like cerebrovascular disease or leukoencephalopathy, of which there are many types, and CADASIL is only one subtype. So it's been really hard to find out the incidence of CADASIL because there wasn't a good way to search databases for this, because there was no ICD-10 code. And now there's a diagnostic code for CADASIL. Make sure if you are a patient that your physician knows this and they use this code. It will help us in research and being able to identify patients for treatment trials and for other studies. I strongly encourage people to contact different CADASIL support groups in your country, one of which is cureCADASIL, but there are others as well. You can find these on different search engines, and to become involved. Just a little plug here for my textbook. This is a textbook that's probably written more for physicians and medical personnel, nurses, people in training. And this is a textbook that I feel is pretty readable. And it's for non-Alzheimer's and atypical dementias, which would include CADASIL. If you notice the two images in the center of these four images, you should have recognized that these are two of my patients with CADASIL who are on the cover of this textbook, the anterior temporal lobe involvement. So some of my CADASIL colleagues contributed a very nice chapter about CADASIL and other white matter diseases that can cause atypical dementias in this textbook, which is published by Wiley Blackwell. So CADASIL is a genetic disorder. Diagnosis can be difficult. It can be confused with many other disorders, both genetic and non-genetic. But the most common diagnosis that it's confused for is just, this should say, I'm sorry, cerebrovascular disease due to cerebrovascular risk factors. We recommend patients be seen by a neurologist, ideally somebody who's familiar with CADASIL or other neurological genetic disorders. Ideally somebody who knows strokes, who knows cognitive impairment. We recommend genetic counseling for all patients and families. I recommend avoiding blood thinners and brain angiography. So invasive brain angiography is usually contraindicated for the reasons we've discussed. And many of the symptoms of CADASIL can be managed, as I've tried to show you, but unfortunately, we don't have a definitive way to slow, stop, or cure the disease. But management of patients with CADASIL, it takes a village. It takes loved ones. It takes community to really help with this disease, as it does with many other neurogenetic disorders. Through research, we hope to find a cure and cure CADASIL. This is a picture of our dementia group at UCSF, Department of Neurology, the Memory and Aging Center. We're probably with the largest group, one of the largest groups in the country, if not the world, studying dementia, including diseases like CADASIL and other disorders. Thank you.
Thank you so much, Dr. Geschwind, for this helpful presentation for CADASIL patients and families. We all know there's many challenges to managing CADASIL, and your experience and insights are so helpful to share with the CADASIL community. CADASIL Care is made possible by volunteers of cureCADASIL, generous donations from CADASIL families, and a grant from Global Genes. This concludes our presentation. Thank you for joining us.